Hello, everyone. Over the last several lectures, we've followed pathogens through the early stages of infections. We've talked about how microbes attach to host tissues, invade the body, and avoid immune defenses. But surviving inside a host is only part of the story. A pathogen can be present in the body and cause very little disease unless it has a way to damage host tissues or disrupt normal physiology. One of the most effective ways microbes accomplish this is through toxin production. Toxins allow pathogens to manipulate host cells from a distance. Rather than physically destroying tissues themselves, microbes can release molecules that alter how host cells function. Some toxins shut down protein synthesis, some interfere with nerve signaling, others disrupt ion transport or activate powerful immune responses. Today, we'll begin by focusing on one of the most common strategies, and that is for the production of A/B toxins. As we'll see, these toxins all follow a similar strategy. They gain entry into host cells and then modify critical cellular processes. The symptoms that develop depend entirely on what cellular targets the toxin attacks. And for this lecture, we are going to get away from our UPEC, Neisseria, and Streptococcus and start going into a couple of different organisms that have nice examples of these types of toxins. So by the end of this module, you should be able to differentiate exo and endotoxins and describe the general mechanism of AB toxins and explain how AB toxins disrupt normal host cell function and produce disease. So at this point, we have talked about how exposure can lead to, or encounters can lead to the establishment of infection by adhesion and then invasion. And then that invasion can lead to colonization if they're able to evade the immune system. Um, so, how do these things actually cause damage, though? We talked a little bit about it with Streptococcus pyogenes, but when we were talking about Neisseria and when we were talking about E. coli, we didn't really talk about cell damage. They're just kind of inside the cell. We're going to go on and talk about these different mechanisms for causing cellular damage, and we're going to start off really with our AB toxins. Um, when microbes induce cell damage, it leads to structural or functional disruption of host cells caused by that microbial activity. Damage doesn't always mean cells are destroyed. Sometimes pathogens kill cells directly. Sometimes they don't. Other times they just alter the normal cellular function, disrupt signaling pathways, or interfere with metabolism. Many of the symptoms we associate with infectious disease ultimately result from this cellular damage, though. Pathogens can damage hosts in several ways. Some produce toxins that directly interfere with cellular processes. Some trigger inflammatory responses that inadvertently damage healthy tissues. And some compete with host cells for nutrients. Others physically invade tissues and destroy cells during growth and spread. Today, we're going to focus on toxins that are being produced specifically. So a toxin is any microbial product that alters host cell structure or function. The important phrase here is alters host cell function. Toxins don't necessarily kill cells immediately. Many toxins work by changing what a cell does, interfering with signaling pathways, blocking protein production, or disrupting communication between cells. As we'll see, even small molecular changes can produce dramatic disease symptoms. Um, the distinction between endo and exotoxins is one of the most important in microbiology, especially in pathogenic microbiology. Exotoxins are proteins actively produced by bacteria and released into their environment. Because they target specific molecules or cellular processes, they are often extremely potent. They can be produced by both gram-positive and gram-negative bacteria. Endotoxins, on the other hand, are completely different. Endotoxin is not secreted. Instead, it is part of the outer membrane of gram-negative bacteria and consists of lipid A within the lipopolysaccharide, or LPS. Rather than targeting specific cells, endotoxins trigger broad inflammatory responses. Today, we're focusing on exotoxins. We'll return to endotoxins in the next lecture. So, one of the most common toxin designs found in the microbial world is the AB toxin. It contains two functional components. The B subunit stands for binding. Its job is to recognize and attach to a specific receptor on a host cell. The A subunit stands for active. Once inside the cell, it carries out the toxin's enzymatic activity. Although individual AB toxins target different cellular processes, they generally follow the same sequence. First, the B subunit binds a host receptor. Second, the toxin enters the cell through endocytosis. Third, the A subunit gains access to the cytoplasm, usually after being cleaved. Finally, the A subunit modifies some host protein, and the result is disruption of normal cellular function and ultimately disease. All right. And so there are many examples of AB toxins. There are also other types of exotoxins. This is just one of the most common types. And so I'm going to give you one example from the microbial world to kind of see how this actually works. So let's look at this example. This is the diphtheria toxin. It's actually encoded by a bacteriophage carried by Corynebacterium diphtheriae. The B subunit is going to bind receptors on the host cell, and the toxin is then internalized through endocytosis. Specifically, this is receptor-mediated endocytosis. Inside the endosome, acidification triggers a conformational change, so that means it changes the shape, that allows the active A subunit to enter into the cytoplasm. The critical step is what happens next. The A subunit permanently inactivates a protein called elongation factor 2, or EF2. EF2 is essential for protein synthesis. So this is involved in, translation. EF2 Without EF2, the cell can no longer produce proteins. Eventually, the cell dies. This illustrates the power of AB toxins. The toxin doesn't destroy the membrane or physically rupture the cell. Instead, it targets a single critical protein and shuts down an essential cellular process. And those cellular processes can be varied. It can cause cells to begin to have an ion imbalance. It can cause cells to rearrange their actin filaments. It's very dependent on the specific AB toxin, but the mechanism by which it gains entry and then is activated tends to be very similar. All right, so the major idea here, exotoxins are secreted proteins that alter host function. Endotoxin is fundamentally different because it consists of lipid A from gram-negative bacteria and primarily causes inflammation. AB toxins share a common design. The B subunit binds and delivers the toxin. The A subunit performs the damaging enzymatic activity. Diphtheria toxin provides an example of how a toxin can produce disease by targeting a specific cellular process, in this case, protein synthesis. As we move forward, you'll see that many toxins use the same basic strategy. What changes is the specific target inside the host cell. With that, thank you so much for attending this lecture, and I hope to see you at the next one.